Hi, you all are once again welcome to BioLibrary. I am Chetan Kumar, your facilitator here at BioLibrary. In this session, we'll discuss about the topic what is double fertilization and triple fusion. Now, remarkably, this is a process which is only seen in the plant kingdom. Normally, all the other organisms have only one single fertilization process and which then gives rise to an embryo or a zygote. But in plant kingdom, there are two fertilization process occurring and which is further helping in the process of sexual reproduction. We shall see in detail what does this double fertilization and triple fusion mean. Here is one of the simplest definition of double fertilization, which says that it is the fusion of one male gamete with the egg cell to form the diploid zygote, and this process is called as syngamy. The second process or the second fertilization which occurs here is the fusion of the polar nuclei of the central cell in the embryosac with the second male gamete to produce primary endosperm nucleus or PEN. This is called as triple fusion. Why is it called triple fusion? Because there are three nuclei which are involved in this fusion, two of the central cell and one of the male gamete. Hence this process is called as triple fusion. Combined syngamy and triple fusion together are known as double fertilization. We shall see in coming slides in more detail what this process actually is. To understand what is double fertilization and triple fusion, we need to go through the sequence of events which an pollen grain undergoes once it is pollinated, that is once it is released from the anther. We know that in majority of the species, that is 60% of the angiosperms or the flowering plants, the pollen grains are shed in 2-cell stage, while in some 40% they are shed in 3-cell stage. However, whatever may be the number of cells while they are shed, I 2 or 3, they land on a receptive stigma on which they germinate. We'll see the details of this germination of pollen grains on stigma in the coming slides. When the pollen grains are released from the anther, the mature pollen consists of two types of cells. As you can see here in diagram, which consists of a vegetative cell and a generative cell. This mature pollen grain, when falls on a receptive stigma, there occurs an events of interplay between the pollen and pistil, and this interplay of pollen and pistil results in the germination of the pollen tube and the further subsequent events of fertilization in the embryo sac. Now what happens exactly here is the mature pollen grain, which consists of the vegetative cell and the generative cell. Here vegetative cell acts as the tube nucleus which germinates into the pollen tube on a receptive stigma. The stigma contains certain sugary substances which enable or favor the growth of the pollen tube. Once the pollen tube starts growing, the generative cell moves into the pollen tube and it starts migrating, as you can see here. On its way, this generative cell divides to form two male gametes. This is possible by the division, which is mitosis, where the haploid generative cell divides again by mitosis to give rise to two male nucleus or male gametes, which continue their path through the style. Again, certain sugary substances in the style also are known to help or attract the pollen tube and favor its growth down into the ovary. Some of the artificially germinated pollen grains are also shown here. When these pollen grains are put into sugary solution, they have a tendency to germinate and the pollen tube can be seen, as you can see here in these two diagrams. The pollen tube, while its way of growth through the tissues of stigma and style, secretes enzymes that digest the surrounding cells and tissues in the style, which further paves the way for the growth of the pollen tube and reaches the ovary, wherefore it helps the male gametes to fuse with the egg cells or the cells in the embryo sac. The pollen tube, while its way of growth into the ovary through the style, may take several paths in order to reach to the ovary. Now this depends on the type and variety of the plant in which this process is observed. One of this path taken by the pollen tube to reach to the embryo sac is through the micropylar tip region, and this is called as porogamy. As you can see here in diagram, the first picture represents the porogamy, where the pollen tube enters the embryo sac from the micropylar region. On the contrary, if the pollen tube enters the ovary or the embryo sac from the chalazal region, then this method is called as the chalazogamy, which is shown here in diagram B. While if the pollen tube takes an intermediate path, that is in between the chalazal and micropylar region, then that method of entry of this pollen tube into the ovary is called as mesogamy. No matter whatever the path may be taken by the pollen tube in order to reach to the embryo sac, it may be either porogamy, mesogamy or chalazogamy. However, the main sole purpose of the growth of the pollen tube is to facilitate the movement of male gametes so that they can reach the embryo sac and fertilize the egg and the central cell. Here is the diagram of a mature embryo sac, which shows the various different types of cells of it is made up of. In the chalazal cell, you can see three antipodal cells, all of which degenerate after fertilization, while there is a central egg cell which is comprising of two polar nuclei, and which is, by the way, the largest cell found in an embryo sac. And on the micropylar region, you find another egg apparatus, which consists of one egg cell and two synergids. Further, you can also observe filiform apparatus in this micropylar region, which basically facilitates the entry of male gametes into the embryo sac. Now, of this, the egg apparatus, which comprises the egg cell, and the central cell, which comprises of the polar nuclei, are the two different sets of cells which are taking part in syngamy and triple fusion. The events that follow in double fertilization are the pollen tube, which reaches the embryo sac, inside which containing the male nuclei. They start dividing and two male gametes are formed inside the pollen tube. This one male gamete will fuse with the egg cell by the process which is called as syngamy and form an diploid zygote which develops into an embryo. Further, the second male nucleus, which is found here in the tube cell, fuses with the already two polar nuclei which are found in the central cell to form an triploid primary endosperm nucleus, and this process is called as triple fusion. As syngamy and triple fusion occur in an embryo sac, both of these fertilization together are known as double fertilization. The central cell with the primary endosperm nucleus develops into an endosperm. The basic function of endosperm is providing nourishment to the developing embryo. So this is the entire process of double fertilization, which includes syngamy and triple fusion. I hope this topic is clear. Thank you once again, and we shall meet in another session with another topic. Thank you.